Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 144th seminar, and I call it a Senti Bioscience Day. Happy Halloween. I mean, today, Halloween in United States, and also I believe some other countries start to celebrate, including Korea. I have been enjoying the sunny San Diego for 3 weeks, thanks to multiple conferences here, including this week's AICHE, the premier chemical engineering meeting. Great to see many friends, colleagues, and also my advisor, Kristalla Preder here. I will embark on a long trip this weekend, including Europe and East Coast for the almost 3 weeks, starting this weekend. Okay, it is my tremendous honor to introduce very briefly, I'm not sure I can do that for this pioneer speaker, our pioneer speaker today, the Dr. Tim Lu. He is now the CEO of Senti Biosciences, but most of us got to know him as a pioneer and also a professor at MIT, and more importantly, a visionary. He has published many big paper, Nature journal paper, etc. per year for more than, I guess, a decade, with all very creative and pioneering new technology development and some discovery. I luckily, got to know him more than 10 years ago when I was a postdoc at the CRISPR lab that was next to his lab. He has been trying to translate synthetic biology technology into a real world product, which we desperately need to demonstrate ASAP to gain the public acceptance of this 2-decade-old field. To me, he is a real genius who I have ever met, inspiring me and many others. And thank you, Tim. Thank you so much for your pioneering work and especially the timely efforts toward translating technology, synthetic biology technology into real applications. And the virtual podium is all yours now. Thank you so much again.
Well, thanks so much, Tata, for the introduction here, and it's very generous of you. I want to thank you for the efforts that you've been doing, continuing to support this community and amplify work that we're all doing together as part of people in this field. I'm very excited to be here today to share a little bit about what we're doing here at Senti, and certainly excited to hear more about what the next generation of folks are doing, including Emil's work later on in this presentation. So I'm going to keep this fairly short and concise, but I thought I would share a bit of what we're trying to do here at Senti in terms of developing therapeutic programs using our gene circuit platform. So just as a bit of an overview, Senti was a company that's based in South San Francisco, really focused on trying to develop gene circuit technologies that can enhance the activity, precision, and control of cell and gene therapies. I think many of these concepts are familiar to folks in this room, but I would love to just give a highlight as to what Senti is doing and why we think this is potentially transformative for cell and gene therapies. In a nutshell, what we're focused on is how do we develop cell and gene therapy products that have greater precision? And so, for example, we can design logic gates that can recognize multiple targets in the body and use that information to better discriminate disease cells from healthy cells. And one of the areas where we think this is going to be really useful in is for cancer therapeutics, where traditionally people have relied on just looking for a single target that's on the cancer, but not on the normal tissues. There's only so many of those targets out there. And for the vast majority of diseases, we need to actually be able to recognize a signature of disease rather than just a single marker. And so leveraging, for example, activating and inhibitory CAR receptors that we can put into CAR T or CAR NK cells allows us to do this logic gating behavior. Number two, as many of you know, a lot of the therapeutic areas that we're going after, like cancer, autoimmune disease, are complex diseases that involve multiple pathways. Being able to simultaneously target those pathways together at the same time and not just hit one target at a time is important. And that's what we call our multi-arming technology, being able to use a single cell to deliver multiple payloads. Number three, many of the drugs that we have in the clinic today would really benefit from greater control over timing or location. And so we have technologies like the regulator dial, where we can use small molecule drugs, FDA-approved drugs, to titrate up or down the activity of a cell and gene therapy, even after it's been delivered into the body. We've also developed technologies like smart sensors, essentially promoters or sensing technologies that allow us to determine where and when this particular product is active in the body. So it's been a many-year journey here at Senti to try to bring these technologies into the clinic. Senti 202 is our lead program, and I thought I would spend a little bit of time there just to show you how we believe synthetic biology can impact the field of cancer therapeutics. But Senti 202 is in the clinic. We do anticipate our first data readout for this product before the end of this year. This product is designed to go after a disease called acute myeloid leukemia using our logic-gated technology platform. So just as a brief overview of AML and why it's important, we're going after a disease called relapse refractory AML. These are patients that have failed frontline therapy and have relapsed. Unfortunately, these patients have a very short median survival, about 5 months or so. Many patients who are initially treated with AML relapse, unfortunately, due to the regrowth of their disease. And currently, the only real treatment for these patients that's thought to be curative, or at least have some good data associated with it, is a stem cell transplant. One of the key biological problems that we believe that it's important to target is the presence of a certain cell type called the leukemia stem cell, or LSC. These stem cells, if you do not actively kill them during treatment, correlate with poor response and poor prognosis due to relapse. And so the idea here is, patients may come initially, present with a mixture of AML blasts, shown here in yellow, as well as leukemic stem cells, shown in red. Traditional therapies like chemotherapy, venetoclax, do not selectively kill leukemic stem cells. And so if these survive post-treatment, they basically can re-go and repopulate the tumor. And there's a lot of clinical data showing that the chance of relapse increases significantly if you have these LSCs present. Fundamentally, one of the key challenges is none of our current drugs explicitly target these LSCs. So what we're doing here at Senti is we've actually designed a cell therapy product. This is using natural killer cells. So this is a type of immune cell derived from the blood of healthy donors. So we get healthy adult donors that come in, they give us their NK cells, and then we genetically modify them with 3 different elements. The first 2 elements are really core to the gene circuits I mentioned before. The first one is what we call an activating CAR. So this is a CAR receptor that recognizes 2 different targets, CD33 as well as FLT3. Both these targets were chosen because they're actually well known in the AML space. They've been validated clinically speaking. However, the reason why we put these 2 together is that they're actually very complementary with each other. CD33 is expressed on the AML blasts, and FLT3 is very well expressed on the leukemic stem cells. So by being able to target both of these targets, we should be able to increase the overall killing activity of AML blasts and leukemic stem cells together. This seems like an obvious idea, so why haven't people done this in the past? Well, it turns out that in AML, as well as many other cancers, these targets are not just expressed on the cancer cells, they're also expressed on healthy tissues, for example, on healthy hematopoietic stem cells. And obviously, killing those hematopoietic stem cells is not a great idea. So what we did here was actually design a second element called the inhibitory CAR, shown here in purple. We did a lot of protein engineering to build in an inhibitory signal transduction pathway that works very quickly, on the order of minutes, to distinguish healthy versus cancer cells. And this purple receptor recognizes a target called endomucin, or EMCN, which is selectively expressed on the healthy cells, but not on the cancer cells. So this acts as a brake for the product. When it recognizes those healthy cells, it basically says, don't kill those healthy cells. Finally, the third element here is an element called calibrated release IL-15. IL-15 is essentially a growth factor that's important for the NK cells themselves, as well as the ability to stimulate surrounding immune cells. So we're really excited about this product. We learned a lot along the way in terms of how to manufacture, how to go through FDA and get this into the clinic. We are executing our phase 1 study now and expect to release initial clinical data before the year end. I'd only have a little bit of time to give an overview here, so I thought I would end by showing you the concept of this product and just briefly mention one other thing, which is how we're running the clinical trial. So the way this clinical trial is designed is to go after relapsed refractory AML patients. We have 2 different dose levels that we're starting with. The first is 1 billion cells per dose of CAR-positive NK cells. And then we have the ability to dose escalate further, for example, to 1.5 billion. If we're successful, we can dose expand, which means we can go into additional AML patients, or there's other related diseases like MDS, myelodysplastic syndrome, where we can go into with this product. So the last thing I'll mention, it's been a really fun experience, really thinking about how to bring this product into the clinic. In terms of the phase 1 trial design, we are using a lymphodepletion step. So what you do is initially treat the patient with a chemotherapy type regimen so that you can open up, quote unquote, some space for the NK product. We're using a particular lymphodepletion called Flu-Ara-C. So that's shown here, before we dose the cells, there's a 7-day time period where they get this lymphodepletion. We can repeatedly dose Senti 202. So at day 0, day 7, and day 14, we give the patients doses, and then we assess at 28 days. We're really excited about the enrollment here. We're seeing a lot of interest from the clinical side for this novel product, and we look forward to trying to release data before the end of the year. Hopefully, as we do that, you guys will hear about it, and we are fingers crossed that this becomes one of the programs that highlights the utility of synthetic biology and logic gating in the therapeutic area. Our goal really is to demonstrate that this product profile does something that existing drugs like small molecules, like protein therapeutics cannot do, something that's uniquely enabled by the gene circuit, which is this logic gate technology. So if we're successful, certainly we hope that this will have an impact in AML, but also in a large number of other diseases like solid tumors, like an autoimmune disease, where you need this greater ability to separate disease from healthy. So with that, I will stop my presentation and turn it back over to Emil.
Thank you so much. It's amazing. I'm thinking about this field, many different way, but I hope, let's say 5 to 10 years later, when it comes to, we think about synthetic biology, we think about Senti Bio, like people think about Gates, Bill Gates, whenever they talking about computer science field. Or the other thing I want to tell you is, or tell the audience, basically, if you have idea, be brave and do it, because we need, someone like you, ambitious, passionate, and then hardworking people jumping into the synthetic biology, develop new product. I believe many of you also have many passion regarding, disease, curing disease, because we all have some beloved people, we lost because of disease. Or if you're more towards sustainability, I believe Emil talking about sustainability, because we also need to solve the problem like, climate change or crisis or waste, whatever. So whatever you wanted to do, jump into the industry or academia, do some more in that way, making the world a better place to live for us and for the future generation. So Tim, thank you so much. This is wonderful. I hope to see your great news at the end of this year or very soon in the future. Thank you.
Thanks so much. Fingers crossed.
Thank you. All right. Now, the main speaker of today with a longer introduction. Dr. Emil Jensen is a tenure-track researcher at Technical University of Denmark, or called DTU, since 2022. He completed his undergraduate studies at University of Copenhagen in 2015 before coming to DTU to pursue his PhD in yeast synthetic biology. During his PhD training, he conducted research at Chalmers University of Technology, Sweden, and UC Berkeley, Joint Bioenergy Institute, or JBEI, USA. Emil is now co-PI, or running independent group at the section Synthetic Biology Tools for Yeast at the Novo Nordisk Foundation Center for Biosustainability at DTU. Emil targets a wide range of applications with his tools, which usually extend from biosensor development in yeast. I met him, I believe, when I visited DTU to give a seminar, and we had a great time discussing many scientific areas and also life. And also, I believe he's passionate about sustainability, or something else, or included. And I believe he has huge potential to become a world leader in his field. So Emil, thank you so much for your time today, and please take it away. Looking forward to your talk. Thank you.
Thank you for the kind introduction. Say so. I'm going to just go into the presenters mode here, and then I think I am ready. You do see my first slide now. Yeah.
Yes.
Thank you.
Thank you.
All right. And of course, thanks for inviting me here to contribute to your webinar series. That's also a good opportunity. So today I'll explain about the synthetic biology in baker's yeast and how we use that for screening in various contexts of biology, basically. But first, perhaps on a lighter note, let me just give you some brief insight as to where I'm currently situated, both in general and also during this talk. I just want to present basically where you might be able to find me in the future as well. So this is Denmark, and it's a small country with 6 million people. And apparently that is equivalent to approximately 0.07% of the world population. But if you want a really good work-life balance, you actually might want to consider going here at some point. So Denmark is also home to many enterprises such as the Novo Nordisk, and the Novo Nordisk Foundation actually founded and still supports our research center here at the DTU campus, which is just outside of Copenhagen. So this is where I'm based right now, and I'm focused in my research on applying synthetic biology tools for yeast. And I'm doing that with my wonderful team of PhD students and postdocs, which you see here on the right. And they're photographed here this summer on a very temperate day in Copenhagen. So now shifting gears, let the fun begin, you might say. So first, a few examples on the projects that we are running in my team. I think that would be appropriate to start out with that. So we mainly deal with yeast-based biosensors development, and these we apply to different topics, and often as part of interdisciplinary collaborations. For example, in one study, we isolate honeybee receptors, olfactory receptors, for signaling in yeast to help decipher the complex social behavior in the honeybees, but from a molecular point of view. We also experimentally model and investigate cell-cell communication in soil fungi, and we do that to make a more sustainable crop management strategies, basically. So this collaborative project also looks at co-occurrences in soil microbiomes and how it correlates with crop health based on satellite imagery. So that again, is an interdisciplinary project. We also work with cell factory development. So we recently published a story on autocrine cell factory screening and selection in double emulsion droplets using FACS. And in another story, we actually activated a surface membrane receptor class called GPCRs in the probiotic yeast Saccharomyces boulardii. And we are now making disease-responsive platforms out of that in another project. And then finally, as an example here, that I will also dive deeper into later in this presentation, is the cancer simulating yeast cell project that we call SCASA. And basically, it is meant to facilitate the studying of phenotypes in human immune cells resulting from co-cultivations with these yeast cells that we engineer. So that's basically the projects that I decided to highlight here today. And the two projects that I'm going to dive a little bit deeper into are listed here. So one is headed by PhD student Giovanni Cesaro here, and the other one by recently graduated PhD student and now, well, postdoc Marcus Deichmann. So I'm going to give you some background on G-protein coupled receptors, the ones I mentioned before, as called GPCRs, because they are a very important class of receptors, both in the human genome. We have more than 800 members of those, and they also make up a substantial part of all the drug targets that are FDA approved. The funny thing is that many of these GPCRs from the human genome, they also function in yeast. I think right now we are about 70 out of 800 working, and that number is rising as we progress in the research. So we can make quite useful biosensors for human health, but also for cell factory screening down the line of drugs and pharmaceuticals, basically using these biosensors. So how does a biosensor work based on GPCRs in yeast? It's very simple, really. Here you see we have an input, could be one of the drugs I talked about before. And it binds to GPCR, which elicits a signaling cascade inside the yeast cells. They also natively have GPCRs, but you can replace those for the human ones. And then they give, in return, an output that corresponds to the input, so to speak. And that could be fluorescence, it can be luminescence, or even growth, depending on the applications that you want to implement. And this is what it looks like, basically. On 3 human receptors in yeast, we're looking at luminescence here, and the increase in luminescence is apparent as you increase the concentration of ligand. So exactly as over here, you have a certain amount of ligand, basically, give the input, and you get a corresponding output. So the more ligand, the higher the output, basically, the more the output. So you can quantify how much is being produced or is in the vicinity of the yeast cells here. So these curves are actually part of a work that was developed here in our team, but also in collaboration with a neighboring team. They're focusing on protein engineering research, and they developed this machine learning algorithm that is really good at predicting drug target interactions with GPCRs. And we are very good at validating that on our yeast platform. So these two going hand in hand is actually on bioRxiv if you're interested. And if you're further interested in a review on how yeast can benefit human health studies, then I suggest you also go and check out the link up here. It came out from our team as of this year, and I feel like we found a pretty good angle on it. It was a fun thing to write, and also I hope that it might inspire some of you at the very least to in your works. But you'll let me know along the way. So over here on the right, there's a lot going on. So this is a Venn diagram. This work was done by Marcus and Jensen in our team previously. And basically, it shows what happens in a wild-type yeast cell or a yeast cell that has human GPCRs during activation of the GPCRs, and how do all of these different strains actually overlap. So 57 genes were differentially regulated in all of these strains, and we use that as a resource to build our biosensors. But the important part here is that for this context, that the wild-type cells, they differentially regulate more than 1,000 genes during GPCR activation. And that leads to mating. And so you can see it's quite a complex process going from, well, let's say a GPCR input, and then all the way down to a binary phenotype that is the mating. So why is mating so important? And I think with that, I sort of took the words out of my own mouth, but I have that in the headline on the next slide. So basically, what we envision with it, at least, is that you can go into liquid cultures in bulk, and if you have like a certain product that you want to screen for, then you can do that autonomously, basically in there, so that the yeasts themselves, they can feel the right partner in terms of what's being produced, or how much is being produced, and then they select that partner for mating. And that is something that you can play it out. So you're funneling down like a ton of possibilities down to fewer possibilities that you can then more qualitatively screen. So in this case, we have 2 diploid cells here of the 2 genders of yeast. And when they mate, they become this diploid cell. And that's basically forming the basis of these colonies that you can then further screen. And GPCRs, they're important, of course, as you can see, for mating in the bigger yeast that we work with as a model. But it's also very important in other communities. For example, fungi in the soil. Many of those also have GPCRs, and they use these GPCRs not only for mating, but like in a lot of different ways to basically feel out their environment. And that can promote commensalism or pathogenicity and so on. But there's not so much necessarily known about that. So what happens down in these fungal communities in the soil, let's say, in crops, so like on the fields, and how is that managed these days? You spread out pesticides, they go into the water and pollute that. So now you hear how we are going into a more sustainable aspect of things, right? So maybe what we would really like to promote with our research is a little bit less or completely going away from all of that, and then towards what you can consider healthy soils. So like new pest management solutions, new ways of basically performing smart farming. But the challenge here is that all of these interactions that are going on between thousands of organisms, not just fungi, but of course also bacteria and so on, they are very hard to study. Many of these organisms won't grow in culture in the lab, so you can't really study them in this way. And you can take them out individually, perhaps, for some of them and studying them. But what happens when they're together, so to speak? So if we combine some of the tools that we have now for searching in bulk, so to speak, with some of the components that we can isolate from these soil fungi, maybe we can actually move towards some of these better, more sustainable crop management strategies. And that is a project I was highlighting before, led by Giovanni. So basically, we are focusing very much on the interaction here between different fungi. We're using a model, Yeast or Saccharomyces cerevisiae. And we take the fungal GPCRs from the soil, basically, those GPCRs, put them into our model, and then we have an isolated focus on the GPCR level, what happens here with this component, and can we devise new strategies for basically controlling the cell-cell communication that is based on these GPCRs. And that, of course, is something that we don't entirely invent from scratch. Fortunately, other people. So this is work from Sonja. I believe she's on next week on the speakers list, I noticed. So that's very neat. She published this work in 2018, where basically a yeast cell expresses a pheromone. So these GPCRs, they often will bind a pheromone, of course. And then the GPCR of a certain soil fungi, of fungus, was then expressed in another yeast cell. And then they do this force reaction matrix, so to speak. So you would expect to see mating all the way down here. Oh, sorry, not mating. This is just a biosensing output. So basically you have expression here, and then you have a biosensor with a fluorescent output up there based on that input. And you see a lot of cross reactions here. And the reason why I said mating is, of course, because now I'm in that line of thoughts. And that's what Giovanni did. So this is just showing some of his data. And there's much more in the bioarchive publication we have out now. But just to summarize here, we also see that individual species, they mate, in this case, with their corresponding partner. And you also have some cross-species interactions. And those are the ones that are quite interesting, we believe. So just to see if there was really like any hold in this in real life, Giovanni, he went on to conduct a chemotropism index assay. So he took the spores of this organism, and then he challenged it with like had a negative control, a scrambled pheromone, its actual pheromone, or a pheromone from another species, so the Fusarium oxysporum, the one that is here. And based on how these spores grow, it was apparent that there was a significant difference in the chemotropism of those, depending on the pheromone. So it seems like indeed there is like communication going on between these species down there in the soil, possibly. So with that, we were encouraged to move on, becoming a little more bold in our endeavors. So basically going towards some of this multiplex screening that I was alluding to before. And for that purpose, Giovanni, he devised a strategy where you have a barcode in one cell, in one strain, and you have another barcode in the other, the one with the GPCR, for example. And then after they mate and become one, these barcodes, they recombine based on a recombinase that's being expressed. And then you can quantify basically how many types of diploids do you have and how big of a proportion do they constitute in an entire population. So like you can basically quantify the output from a mixture based on quantity PCR down the line. So we looked at this interaction network, you can call it a pH 6. And basically you'll see that this pheromone here, that's how we denote it. Like we have phero for pheromone, and then we have ST2 for the GPCR. It can also be called HPR2 depending on the organism here. Then we look at how they actually communicate with one another. You can see this GPCR strain would mate with this pheromone producing strain, but not as much as with its actual cognate partner, so to speak. So now we have like a map of what's of what it might look like when you have individual fungi, but in a complex mixture. And then Giovanni asked, so if you change like on some abiotic factors, for example, pH, which may vary in soils, then how does that change the interaction that works in the cell-cell communication between the fungi? So going towards pH 7, that's what you call slight change, is very apparent in its difference is very apparent in cell-cell communication, right? Because all of a sudden you have a much larger proportion of the interaction over here in the Fusarium oxysporum. You have some diminished interactions which were there before. So there's like some interactions that change evidently solely based on the pH and at the GPCR level. So that's also very important to remember that we're actually studying a single component of a much more complex system here. And that's the beauty of it. All right, so now that this was established, we have this model over cell-cell communication networks from soil fungi. Can we tune that in smarter ways than just like changing the pH, let's say? Can we actually deliver something potentially in the future to control these communities, to promote commensal yeast, commensal fungi, and vice versa with the pathogenic ones? So Giovanni, he set out to screen a library of pheromone variants against one GPCR strain. And he did that for a number of GPCR strains, basically with the same library. And after this nice workflow, what he gets out of it in the end is a genome extraction, which can then be sent for Illumina sequencing. And then he looked at which hits were most enriched, basically, for finding new agonists that work even better on these GPCRs that we were looking at before. But just a little more about the library. So there are some constant parts in this library, and then there are like some interchangeable parts, so that we maintain like not an astronomical size here in the beginning. It's proof of principle, and it gave 8,000 variants at the amino acid level, and 250,000 of variants at the DNA level. And because we delivered these libraries on plasmids, we want to make sure that we had a high efficiency of transformation. And we used a new protocol, also from our team, developed by Markus Vinskog. You can find it here. So basically, it increases the yield of plasmid transformations 100-fold. So that's quite neat that we were able to use it already here. So what happened here after Giovanni looked at the most enriched variants in his screen from the sequencing data was that he found two variants called A4 and A5 of the native pheromone, here shown in comparison these dose response curves as we saw previously as well, that actually respond to lower concentrations of these new variants. Like they make the receptors respond to lower concentrations of these new variants as compared to before. So that's quite interesting that you can actually find maybe new biological solutions to interfere with the communication networks there. And what was also really interesting was that if Giovanni looked at the beginning, like the least enriched hits, he found something that would be an antagonist. So a pheromone from this C. militaris worked on another receptor as an antagonist as you go up in very high concentrations. But still, nevertheless, very interesting findings from this soup, so to speak, of possible interactions. So being quite amazed with that, he went on to test what he had found in in these communication networks again. Now we're back to the quantitative PCR for finding that out. And I'm just going to show you like all the plots here all at once, but there is some explanation to it. So up here, we are looking at interaction networks. The individual abundances of diploids is being shown over here. And we are using both a scrambled pheromone, so one that's not supposed to do anything in there. It's just to see like does the mere presence of a pheromone do anything. Then the antagonist and one of the agonists that were found. You can see that there are clear differences just by looking at these diagrams. But you also see for the individual interactions of a GPCR with a certain pheromone, the distributions are different here as compared to down here, as compared to down here. So there's a lot going on from the same assay, but just by supplementing either an antagonist or an agonist down there to change the interaction networks. So that was super neat. And then I just want to like pick up on all these fragments that are presented here, because it is an ongoing work now the platform is developed. But of course, we want to elaborate much more on it. So just to briefly recapitalize here, we have this one-on-one, so to speak, interaction network with mating as a readout. We have one versus many, as we call it, where we can basically screen libraries of pheromones for, well, new functions on their, or improved functions or so on certain receptors. And if we spike those hits into cell-cell communication networks, then we get these controlled distributions over here. And you can also see the replicates that is quite a robust technique. So it's not like it's all over the place. It's actually something that's reproducible. Something I didn't show so far, but I just want to leave off with this basically, is that when we spike in urea into the media of the yeast cells here, we see sometimes significant differences in these distributions that we were also looking at up here, but not for the negative control and not always. So some are more robust interaction-wise than others. And urea is, of course, like a heavily used fertilizer. So it's interesting that we might be able to actually pick up on different use of fertilizers and how they impact these cell-cell communication networks on a GPCR level. And so that, of course, leads me to the very last point in this slide for this story. In this collaboration where Matis is doing the satellite imagery analysis part, him and Giovanni has collected a lot of soil samples from different fields here in Denmark now to basically get better resolution, more data, to investigate much better what's actually going on down there and how that might be impacted by what we do up here. So that's, of course, what we are working towards these days. And in the end, hopefully some of this will be able to actually alter the way that we do pest management and have better sustainable agriculture. Okay, I just want to also present one more project here in depth, as I promised in the beginning. So this project is quite different in many ways, but it's also in some ways the same. So Marcus, leading this project, she previously worked with me on yeast mating and the mating story. You saw his picture before as well. But he wanted to take what he learned about GPCR signaling, basically converting the input signal to a graded output signal, in this case to antigens that can be recognized by human immune cells, which then also the phenotypes, and that we can characterize. So you can see how now we're doing some really interdisciplinary synthetic biology. If I want to explain a little bit more about this graph here, we have many modules, so to speak. We have the biosensing part. The tunability is very important. Also, if you are to model different cancers, you need to also be able to control the antigen densities on the surface of yeast. So we are combining that technology with the GPCR activation. And then there's, of course, the whole binding, synapse formation, so on, and the communication with the interspecies communication, so to speak. And this is what it looks like. It's an early picture that Marcus took of yeast cells, these gaser yeast cells in combination with CAR T cells. And you see the interface here. So that's what it looks like in reality. And then we wanted to make sure that yeast doesn't innately, so to speak, activate the T cells, because that would be a problem for this entire co-cultivation study, right? So basically the yeast cells up here, some different strain designs. They would only grow in yeast media, the blue and the green curves here, but they completely flatline in the mammalian media that we used. So that was very nice. They don't overtake the culture. They seem to just be around. Also, the viability of the T cells, naive T cells, didn't seem to drop significantly, at least. So that was also good. And they didn't go completely crazy by unspecific activation from the yeast, as we see over here in the proliferation assay. So all of these things were very good indicators for us to basically carry on with this project, for Marcus to really like power through, because until this point, we couldn't be entirely sure, also based on literature and so on, but we can discuss that further down the line. So empowered and encouraged, Marcus, he said, okay, I need to be able to control the antigen densities on the surface of yeast here. So we first had to take into account like the GPCR signaling and the output. He chose CD19, which is a B lymphocyte antigen, to basically model this interaction between the immune cells and the yeast cells. And the basic principle here is, again, that we can control the antigen densities in a graded manner, depending on the input that we give the GPCR. So what it looks like when we use different pheromone-inducible promoters, that's basically what we're looking at here. You see that some of them are more powerful than others. Down here, you can see like a better distribution. Some of them are pretty tight. Others are less tight, but it comes and goes with different advantages and disadvantages, so to speak. We have a really high fold change, span of really high fold change from on to off in all of these designs. Interestingly, there were also some constitutive promoters that respond well to the pheromone, so got an additional boost, which is something that Marcus also dived a lot into. And of course, you can read much more about that also in the preprint. So basically, what happens when we take these designs and co-cultivate them with CAR T cells, well, in this case, Jurkat cells, it was quite interesting to see how the pathways inside these Jurkat cells were activated, differentially depending on the different designs. We had like a negative control down here, the one called empty. We had a very tight but also not very powerful design here. Then we had some more powerful design here and the most powerful design up there. So we have this really, really big fold change going on. And at the same time, compared to NALM6 cells in all of these settings here, it seems like we have a more powerful design. So that was quite interesting that we actually go beyond using an actual cancer cell. So again, encouraged with these results, can we make a cancer cell simulator and validate cell therapy products? That's the question that Marcus asked, because we want to do something real with this. We want to make an impact. And so he got a little help from Giovanni, who also is now very much into the other project I presented before. But Giovanni does many things. So here he's helping purifying blood that we got from the local hospital, from a patient, which was CRISPR engineered to become so we could get CAR T cells from them, basically. And then we want to see on this cell product that is supposed to go back into a patient in future, then does that actually work? How does it perform? And that's where scarcer yeast cells come into the picture. So I like this conclusion here. The product was functional, but if you compare it, like this is the product that we like. This is scarcer yeast cell that we tested together with NALM6, and then again like this empty negative control. And it's very clear that when you don't have a CAR engineered into the T cells here, there is no activation going on as per the CD69 staining. But when you have the engineered T cells with a CAR, you actually do see that there is a really high change, and that change can also be controlled by, or that activation can really be controlled by just tuning the ratio of target cells to effector cells ratios, basically. So akin to what we did for the GPCR induction, can also be done by tuning the ratios of the cells. But the good thing with the GPCRs is that you don't need more cells, so you don't necessarily take up more resources. And if you ever need to remove the yeast again down the line, it would be nice to not have overly many of those and just control the whole thing by GPCRs. So something that was really interesting here, Marcus noticed, was that the yeast cells, they didn't have the same level of CD19 displayed on the surface as the cancer cells and the NALM6 cells. So how could they then activate more intensely as based on the intensity down here, if you compare those two, slightly more intensive, even though they don't have the same level of CD19 displayed. And the explanation that he came up with was that whereas many of the NALM6 cells, they lose the CD19, so they basically relapse, so to speak. The yeast cells don't, because they are orthogonal. So either the cancer cells, they get killed, or they just lose the CD19 on the surface, so they adapt to the circumstances. We have a more constant level of CD19 display on the yeast cells, and also an amount of yeast cells around. And that's something that he proved over here, where we have a population of NALM6 cells that are all happy. They have a lot of CD19 being surface displayed here, so to speak. Whereas when you add the CAR plus cells, then they lose that. So they're still alive, but they have relapsed, so to speak. So they have lost the CD19. And in comparison, the yeast cells, they seem to be much more robust and stable on this part, which is something that I think would actually more resemble the in vivo conditions. Because like even though a CAR T cell might kill one cancer cell, there's still a ton of others. So it's not like they go away like they actually seem to do in these in vitro assays. They stay constant, I would assume, like on a population level. On that part, I would argue for yeast. All right, and that's basically also what we're trying to show here, that CD19 is lost for the NALM6 cells, but not for the yeast. Okay, we are headed towards the very ending. I think the time is also more or less up, so that's good. The scarcer yeast cells, they have a lot of applications that we envision, both in vitro and in the long run. Would be amazing if we could go in vivo, of course, maybe even help making some of these phenotype-specific production pipelines. But of course, there's much more to be done on that part, and also what's called regulations, regulatory authorities to convince about that down the line. But it has to start somewhere, and the work that Marcus did here is a really, really big step towards that, I believe. So why yeast? Why scarcer yeast? It's very cheap. It's very straightforward to use, to engineer, and again, it's very cheap. So just keep that in mind. If it performs on par with other assays, which we show for the NALM6 cells and which we're also showing for beta assays in the near future, then it's a really good and cost-effective alternative. So now we are at the last slide here. A lot of names, everyone contributed. Really happy with the collaborations that we have going on. This is the team as of right now. I think Trine went out here, but we have quite a good pool of young faces here. And I would like to thank everyone who has contributed to funding our research in the current team as it is. Oh, extra slides. That was the last slide. Thank you for your attention.
Amazing talk. I like it. So you are doing many different projects, it's including the CAR T cell that is also relevant to the team news. Unfortunately, the team need to leave because of another meeting. So let me check whether we have the question. So let me start with some questions. By the way, I love the connection between you and Sonia. I think the next week's speaker. And this is how we connected. It's kind of randomly kind of schedule, but somehow connected back to back. That's kind of amazing to me. So the question is, you kind of show the power of yeast. I'm not yeast person. Actually, we for the first time preparing one paper that's about also yeast sensor, but not the sensor like using your kind of system, but more about the RNA sensor. So we are we just try to finish. Actually, we finished the experiment. We try to finalize the draft. So I'm pretty fascinated by the power of yeast. And then yeast has been, powerhouse or real commercial product, generator for many, many years. Of course, the famous example is we drink alcohol that coming from, some yeast, also some other thing as well. Uh, so people, sometimes said, yeast we know a lot, but we still don't know, many other things. So to make your technology, your sensing technology and your some other kind of the technology today you show to go really one step further from the just traditional fermentation to biosensing application or something else. What remaining challenges we need to overcome? Because I mean, some people saying, "Oh, it's easy," but that's not true. I mean, still we have a lot to do, right? So I just want to hear from your perspective or your insights. I just could you share that with us?
Yes, definitely. So like it's more about taking a pick here, right? Because there's so many directions as you have seen that we can go into, and we can't cover all the ground, but we cover quite a lot of ground, I think, in different areas. So it's really a matter of like, where would you like to place your effort? Because yeast is a really good platform for synthetic biology. It's very easy to engineer, and you can do so many things with it. But there's also a lot of unknowns. I think something that's really important to pick up on, at least something in our scope, is how you can use mating in yeast to um, make these crazy selection regimes. Because in my opinion, the real big problem that we are facing is that we can't test everything. If we could build and test everything, we would never have to predict anything, right? We would have perfect models and so on. So like, different ways of approaching the problem. But we can build a lot by now. We can really generate a lot of variants. I think can easily generate like 100 million variants, introduce that into yeast in a high efficiency transformation these days, high yield transformation, I would call it. But then again, like how do you screen more than 100 million variants? If you do that 10 times in a row, you have a billion variants. And how do you screen that? Are you going to use fax? It's going to take forever. I think people overestimate it. So it's really like you want to just take all of this that you have transformed, all these variants, put them down into the soup, and then look at your plate afterwards. You control the conditions, you can make them more astringent. And then when you have this growth-based selection, you're really falling down from billions to a few. So addressing challenges in terms of screening, I think yeast is very powerful. I think it boils down to that because we can screen a lot of different things, drug-target interactions, cell factories, you name it. So it's really a matter of like focusing on the power of yeast for screening, in my opinion.
I see. So that's good point. I still learning about this is because actually when I was in the company I just deal with yeast very briefly, but that's not the genetic engineering at the time. It's more likely fermentation using the already developed yeast strain. We try to make some other thing at the time. And then now, several years ago, I start yeast to develop some sensor, but unfortunately the postdoc left, and then we stopped doing so. And then a few years ago, we actually start again the yeast project, to develop sensor again. And then finally we finished that project. So I'm really glad. Now I'm not just bacteria guy. I'm just also eukaryotic enter eukaryotic, world. I mean, finally. So I'm very glad about that one. Let me check whether we have question. Okay. So let me ask one more question related to that one. So where you want it to go? I mean, next 10 years. I mean, there are many potential application you demonstrated today, including CAR-T, and the many sensor you just introduce, then what is how you envision 10 years from now you would be?
So for myself, that would really be sticking my neck out, right?
Yeah, in terms of technology development or science, I mean, wise, I mean, where you want it to be, I mean, so this technology, 10 years from now, I'm just curious.
Yes, I mean, there are two sides of it, right? There's, of course, like the impact you make with startups. There's also the impact you make in the academic track. And some can do both. Some need to choose where you put your emphasis, right? So some of these technologies that we are working with right now that are represented here,
Mm-hmm.
definitely would be great if they could make a real impact in the end to guide better designs for the different applications, or be used directly in certain, let's say, therapies applications on the fields and so on. So,
Mm-hmm.
like taking it to real impact, so to speak, is something that matters a lot, also to me.
I see. I see. That makes sense. That will benefit a lot of people in the world, I think. So, fantastic. Let me see. I don't see any question now. And then this is already 1 minute before the 11 a.m. time here, not the central time. So let's close and then we probably chat afterward. Okay, so thank you for joining us and staying with us today. We'll meet again on November 7th, Thursday, the same time, the same Zoom link. As I mentioned, we'll have Professor Chimeng Tan at UC Davis, also Professor Sonia Beck, and I think still University of Groningen, but she probably moved to another institute very soon, I heard. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I stopped recording. 1 second. Just 1 second.